Hello everyone, and thank you for joining us for the inauguration of the UMC Utrecht as a FUS Foundation Center of Excellence. My name is Thomas André, and I'm the FUS Ambassador for Europe for the Foundation. We will have a couple of short overview talks of the center and the research projects the groups here will be working on. We will start with Professor Crit Moon, who will serve as the scientific director of the center. He will give an introduction and overview of the UMC Utrecht FUS Center of Excellence. Before we get started, a few technical items. If your connection is lost, please simply log in again through the link you received when you registered. At the conclusion of today's webinar, you will receive a link to the webinar recording as well. If you'd like to ask a question, please submit it at any time via the Q&A function at the bottom of your screen. We will collect these questions and answer as many as possible during the Q&A period at the end of the session. Before we get started, I'd like to ask all the panelists to turn off their cameras. And now I'd like to introduce Professor Crit Moon. Crit, the floor is yours.
Hello, ladies and gentlemen. Welcome to the special seminar on the occasion of the University Medical Center Utrecht becoming a Center of Excellence of the Focused Ultrasound Foundation. The foundation is based in Charlottesville in Virginia, the United States, and has been the key promoter of focused ultrasound and its clinical use worldwide. My name is Crit Moon. I'm professor in image-guided molecular interventions at the UMC in Utrecht, and I'm the head of the therapeutic ultrasound group at Utrecht. So a few words on the program today. We will start with a few animations on what focused ultrasound really is, how it is used at the University Medical Center Utrecht, and with its partners is the Prinses Maxima Center for Pediatric Oncology at Utrecht and the Isala Clinic in Zwolle in the Netherlands. They both play a key role together with the industry partners, as you can see on the slide. Then after that, we will have presentations. We have the excellent speakers. We have the luxury of having excellent speakers from Utrecht, from the foundation, from Europe, from Canada. And they will explain the use of focused ultrasound in medicine as they relate to the program, the research program at Utrecht. Then we will come back to with a question of answer sessions, and please pose your questions for that. After the animations, my colleague, Professor Lenny Verkooyen, will take over. She is the manager of research of our division of imaging and oncology. She's also the head of the trial bureau, and she is the principal investigator of the European funded research project further on the use of focused ultrasound and radiotherapy for pain palliation in patients who are suffering from pain from bone metastasis. That's all as of this point. So I would say let's continue with the animations.
Focused ultrasound is an early-stage, non-invasive therapy with the potential to transform the treatment of many diseases. Much as a magnifying glass can be used to focus on the sun's rays to burn a hole at a single point in a leaf, this groundbreaking technology concentrates multiple intersecting beams of ultrasound energy to precisely treat areas deep in the body without incisions or radiation. This leads to less risk of infection, reduced complications, and faster recovery.
Binnen de divisie beeld van het UMC Utrecht werkt een grote groep mensen aan nieuwe technologie voor het non-invasief behandelen van kanker met behulp van gefocuseerd ultrageluid. De technologie wordt MR-HIFU genoemd en is een mooi voorbeeld van opereren zonder snijden.
HIFU staat voor High Intensity Focused Ultrasound. Bij MR-HIFU is in het bed waarop de patiënt in een MRI-scanner ligt een HIFU transducer ingebouwd. Dat is als het ware een luidspreker waarmee ultrageluid heel precies en gericht de patiënt ingestuurd kan worden. Hierdoor ontstaat er een brandpunt met een erg hoge geluidsintensiteit. In dit brandpunt wordt het weefsel, bij kankerbehandeling een tumor, snel verhit tot wel 60 graden Celsius, waarna het afsterft. MRI-scans laten daarbij precies zien waar het focus geplaatst moet worden en hoe warm het weefsel daar wordt. Het gezonde weefsel rond de tumor kan zo gespaard worden. Klinische behandelingen in patiënten worden in het UMC Utrecht al uitgevoerd van goedaardige myomen in de baarmoeder en uitzaaiingen in bot. Er wordt hard gewerkt aan het mogelijk maken van de behandeling van andere vormen van kanker, zoals borstkanker en tumoren in de lever en de alvleesklier.
Een team van fysici, technici, artsonderzoekers, laboranten en medisch specialisten is hierbij betrokken en het werk beslaat het hele spectrum van techniekontwikkeling tot klinische implementatie.
In nauwe samenwerking met het UMC Utrecht en Philips Healthcare werd er een MR-HIFU-systeem voor de behandeling van borstkanker ontwikkeld. Dit is een uniek systeem dat speciaal werd ontworpen voor de toepassing in de borst. Rondom een cup in het MRI-bed bevindt zich een ring met HIFU-transducer-elementen. Recent is in een eerste studie met dit systeem aangetoond dat het brandpunt nauwkeurig kan worden ingesteld. Met geavanceerde MRI-thermometrietechnieken kan het temperatuursverloop in het weefsel tijdens de verhitting goed gevolgd worden. De volgende stap is een studie waarin gekeken wordt of het mogelijk is een borsttumor geheel door verhitting te vernietigen, zodat in de toekomst voor sommige patiënten chirurgische verwijdering van de tumor niet langer nodig is.
Dit sterk multidisciplinaire werk is mogelijk door de integratie van de betrokken onderzoeksgroepen in het ziekenhuis en de nauwe samenwerking met de industrie. We verwachten dat MR-HIFU kan uitgroeien tot een volwaardige non-invasieve behandelmethode voor verschillende vormen van kanker.
That was a very nice pitch, a nice animation and a good start of this webinar, I think. And it is now my pleasure to introduce to you a very nice range of speakers who will give a short pitch about their research focus in the field of MR-HIFU. I'll do this together with my colleague Crit Mone. My name is Lenny Verkooyen. I was just introduced by Crit Mone, and it is a genuine pleasure to be in this position to introduce this impressive line of speakers. The first speaker is Dr. Neil Cassell. Dr. Cassell is a neurosurgeon and the former co-chair of neurosurgery at the University of Virginia. And Dr. Cassell has contributed tremendously to the scientific and clinical field of MR-HIFU in the brain. He also participated as a Blue Ribbon panel member in the Cancer Moonshot Initiative of tomorrow's new president of the US, by then the Vice President Joe Biden. And Dr. Cassell is also the founder and chairman of the Focused Ultrasound Foundation, and it is really a genuine privilege to announce to you his pitch today. After Dr. Cassell, we will have a short introduction of our division, the Imaging and Oncology Division, by Professor Jeroen Hendriksen, who is a radiologist and also the chair of the division in our hospital. Crit.
And then following Jeroen Hendriksen, our next speaker will be Neil Lipsman. He is from Toronto, Canada, where he is working at Sunnybrook. He is a clinician scientist in the field of drug delivery in the brain, and is really the first author of several groundbreaking papers in this field. So we're really happy to have him as a guest speaker. And then after that, after Neil Lipsman is Dennis van Verde. He is, as we've shown before, he is in the Princess Maxima Center, our neighboring center next door to the UMC Utrecht. He is there in the Department of Neuro-Oncology. He is an oncologist, and he is together with my colleague Mario Rees spearheading the efforts in introducing the drug delivery in the brain.
Okay, and then after Dr. van Vuren, we will have our medical oncologist, Dr. Karijn Suikerbuik, who will give a pitch. She is a clinician scientist at the Department of Medical Oncology and a PI in a wide range of studies, including a study on immune stimulation with focused ultrasound. After Karijn, we will move to Italy, and it's a pleasure to also introduce to you Dr. Alessandro Napoli from Rome, who is a director of the clinical development of MR HIFU units, and also one of the most prominent leading MR HIFU clinicians worldwide. And I have the privilege to work with Alessandro in the Verde project, a Horizon 2020 funded project looking at the use of MR HIFU for bone metastases. And in this project, we also work with Dr. Martijn Boomsma, and it's also a genuine pleasure to introduce him to you today. He is a radiologist at the Isala hospitals in the east of the Netherlands, and he is the director of clinical development of the MR-guided ultrasound, focused ultrasound unit at Isala. And last, but definitely not least, my colleague Manon Braat from the UMC Utrecht. She's a radiologist and a PI in MR HIFU breast cancer ablation studies. And she's closely involved in the scientific and clinical work of MR HIFU in the UMC Utrecht. So we will now hear all the pitches of these speakers after another, and I encourage you to already submit your questions in the Q&A box that you can find in your screen, and so that we can move on after the pitches directly to the moderation session. So enjoy the pitches, and thank you very much for your attention.
Good afternoon. I'm Neil Cassell, the chairman of the Focused Ultrasound Foundation. The University Medical Center in Utrecht is recognized as one of the leading sites for focused ultrasound activities around the world. And we are thrilled to welcome you as our newest Focused Ultrasound Foundation Center of Excellence. The Foundation's Centers of Excellence are luminary sites that are created as a result of multidisciplinary interdepartmental efforts that are able to achieve excellence in patient care, research, and education. These centers are created as a result of partnerships between industry, academia, and the foundation, with the purpose of showcasing the technology and serving as hubs for collaboration. Under the direction of Krit Moonen, there's a long track record of technology development, including things like volumetric ablation and MR thermometry and organ motion tracking, clinical trials of bone metastasis and breast cancer, and patient treatments at the ISALA Hospital and Princess Maxima Pediatric Oncology Center. There's educational efforts that include FUS group monitoring of 19 PhDs over the last 10 years, and the PhD curriculum of the Medical Imaging School includes more than 150 students. So again, we congratulate UMC Utrecht as achieving designation of one of the Foundation's Centers of Excellence. This will broaden the scope of disorders treated, and it will be a driver of clinical adoption for focused ultrasound treatments for patients in the Netherlands and beyond. This will facilitate achieving the Foundation's vision of accelerating the development and adoption of focused ultrasound that will enable improving lives of millions of people around the world with a wide variety of serious medical disorders.
Hello, my name is June Hendriksen, and it's an honor to present the webinar on the occasion of the UMC Utrecht becoming a Center of Excellence of the Focused Ultrasound Foundation. At the University Medical Center Utrecht, we have a focus on image-guided interventions, and we are one of the accelerators of the UMC Utrecht strategy. This is part of a long history we have with also other methods in addition to the MR HIFU. For instance, the MRI Linear Accelerator developments and the technique of radio embolization with, for instance, the holmium microspheres. For these methods and image-guided interventions, we have a complete innovation loop from the first ID to the daily clinical practice in clinical care. And these are developments performed in academic industrial partnerships. To give you a short example of another innovation in addition to the MR HIFU, where you will hear a lot about today, this is the MR Linear Accelerator, where in the last 20 years in our division in the radiotherapy department, there has from a development from the first idea together with the industrial partners to now a working machine that is working throughout the world, and that you will see on the next slide. This is the combination of MRI together with the linear accelerator to really guide treatment. These developments have been performed in close partnerships with, for instance, Philips. And a lot of these developments are on real-time MR image analysis. There have been large investments in the MRI HIFU platform and the HIFU group that started a long time ago with Professor Willem Mali, who really saw early on the high potential of this development, and was continued with Peter Luiten in the last 5 years. In the last year, there have been huge investments from the Princess Maxima Center for Child Oncology, which is really next to our UMC Utrecht, and in close collaboration, we have now an MRI HIFU brain system, and we focus with this technique on drug delivery for blood-brain barrier opening. And later on today, there will be a presentation both from Neil Lipsman from Toronto and from Dennis van Vuurden from the Princess Maxima Center. We have a large range of clinical partners. There is a unique position where we have a combined imaging and cancer division who are both combined in one organizational structure. I think this is a very unique situation where we have really the synergy of the imaging developments and the cancer patients. And a lot of the people and researchers working in this group work closely together, from oncologists to hematologists and others. As I said, we have a lot of collaborations with the Princess Maxima Center, from which you will hear later on today, which is a unique proposition where started a few years ago, where all the child oncology in the Netherlands is centered in one center, which is the largest child oncology center in Europe. Furthermore, we have a collaboration from the HIFU developments with a non-academic hospital, where there is also a similar type of machine for HIFU available, and really active in the field of high field. And Martin Boomsma will give a presentation on that later on. Furthermore, there is a strong European network with the Verde project, where the UMC Utrecht is the leading center with Lenny Verkooyen and Craig Monen. Furthermore, there are transatlantic collaborations with the NIH, Brad Wood, and with, of course, the FUS Foundation. With that, I want to give the floor to all the presenters today, and I'm really looking forward and honored to be part of this Center of Excellence of the Focused Ultrasound Foundation. Thank you very much.
So thank you very much for the opportunity to take part in this terrific event. My name is Neil Lipsman. I'm a neurosurgeon and scientist at the University of Toronto. My brief presentation will focus on focused ultrasound and its disruptive potential, specifically in oncology. And to begin with, I think it's important to reflect on what we mean by disruption. And in this case, we're really talking about a tool that replaces something that is existing or that creates an entirely new market. And focused ultrasound essentially has done that already. So when you look at the number of publications per year in the brain focused ultrasound space represented by this red line, you can see an exponential growth in focused ultrasound research in the last 30 years. And a lot of that has been driven by two dominant publications in the field. One that established that you can use focused ultrasound to transiently open the blood-brain barrier in animal models, and the other one, the first attempt at using transcranial ultrasound to ablate tumors in the brain. So its disruptive potential has already been recognized and has driven a lot of the progress in the field. So the blood-brain barrier applications are really especially promising, and this gives us the ability to use focused ultrasound in a spatially and temporally precise way, in an image-guided way, in a very controlled manner, to, in an awake day surgery type of procedure, open the blood-brain barrier in a very focal way, virtually anywhere we want in the brain. That is the potential of this technology. And what we've now shown around the world is hundreds of preclinical studies ranging from small animal models all the way up to non-human primates that have shown that, in fact, you can use focused ultrasound to open the blood-brain barrier temporarily, repeatedly, and safely in myriad brain regions, ranging from eloquent to non-eloquent brain structures. And what we've done here in Toronto is leverage that preclinical experience, and a few years ago conducted one of the first trials of opening the blood-brain barrier specifically in brain cancer, as it represents one of the biggest challenges in clinical neuroscience and neurosurgery, and is one where the blood-brain barrier is an especially important obstacle for the delivery of potentially effective therapeutics. So in this work led by Dr. Todd Mainprides at the time, we showed that we're able to reversibly open the blood-brain barrier, as demonstrated here with this contrast-enhanced regions immediately posterior to a previously resected tumor, and we can do that in a reversible and safe fashion. And since then, we've expanded this work into a phase 2 trial where we're helping to deliver temozolomide or Temodar in patients with primary glioblastoma who are undergoing maintenance chemotherapy for their disease. And the objectives of this trial is really to build on the previous work and show that in multiple patients, we can do this repeatedly, and we can do it in much larger brain volumes. So this is one example of a patient with a left-sided paramedian glioblastoma previously resected. And this heat map here doesn't represent temperature, but it represents a measure of acoustic emissions that are an indirect measure of opening the blood-brain barrier. And this shows you how you can essentially conform the opening of the blood-brain barrier to much larger volumes. And what we found is that these procedures are very well tolerated. They're done with patients awake. They're obviously non-invasive, and the vast majority of patients are discharged on the same day. So this ability to less invasively open the blood-brain barrier in a highly safe way, in a highly novel way, while delivering a chemotherapeutic drug could potentially really change the way we treat these disorders. Now, we're also interested in recurrent tumors, which are particularly challenging in the glioblastoma population. And here in a phase 1 trial, which is a multi-center trial that is sponsored by Insightec, we're recruiting patients with previously resected and treated GBMs who have recurred and delivering carboplatin to these patients immediately before or after the blood-brain barrier is open. So this is an example of one of the first patients treated on this trial, and you can see a large volume of the blood-brain barrier has been opened, in this case more than 65 cubic centimeters. So you can see a very long way away from the initial work, which opened very small volumes. Now we can open much larger volumes. And again, same-day chemotherapy in an awake, non-invasive procedure that lasts less than 3 hours from the beginning to the end. So in this case, this work is continuing, is ongoing, and continues to recruit, and is a multi-center study. We're especially interested also in metastatic cancer, and in a phase 1 trial, we're using focused ultrasound to enhance the delivery of trastuzumab or Herceptin to patients with known demonstrated HER2-positive breast cancer. So Herceptin is a very large antibody-based chemotherapy that ordinarily cannot cross the blood-brain barrier. So we know that we can get exquisite control of cancer, of breast cancer, metastatic breast cancer, everywhere except intracranially. So the prospect of being able to deliver this chemotherapy to the brain is very important for oncologists, radiation, medical, and neurosurgeons as well. And in the first 2 patients, we've shown that we can, in a safe, reversible manner, open the blood-brain barrier, including in regions of the brain that are highly eloquent, such as the posterior fossa at the level of the medulla, near the ventricular surface and near cranial nerve nuclei. And we can do this in a highly safe, controlled, again, outpatient manner. The next steps for this trial are going to be to add treatments, serial treatments with combinations of chemotherapies. And we're also very keenly interested in labeling, radio labeling the chemotherapy in order to directly visualize its presence in the brain and essentially confirm that we can, in fact, deliver large compounds to the brain across the blood-brain barrier. So in this way, I wanted to just provide an overview of the very growing potential of focused ultrasound purely in the oncologic realm, where it arguably has some of the greatest potential impact. We're interested in using it for primary brain tumors, secondary brain tumors, and in work we're very excited about in collaborating with the group at Utrecht and with your group in pediatric brain tumors as well. And we'll hear much more about that in subsequent talks and in the future. So if we can essentially provide treatment options for patients up front, at maintenance and at recurrence, we can potentially cover the full spectrum of oncology across the board, really potentially moving the dial. Among the most exciting advances in the field is going to be liquid biopsy and the possibility of enriching peripheral samples for things like cell-free DNA, permitting perhaps diagnosis and monitoring of treatment as it goes on. And you'll hear more about that to come. So focused ultrasound, what does the near and far future look like? Well, I think a lot of this stems from its power both as a diagnostic, treatment, and potentially a preventative tool. So we can envisage a future where we can make diagnoses with liquid biopsy perhaps early in the scanner when patients are less symptomatic. We can treat patients to deliver disease-modifying therapies and perhaps improve their quality of life and slow disease and reduce mortality, especially from recurrent disease. And perhaps down the road, use it as a preventative strategy to deliver things like vaccines and neuroprotective agents to ultimately even reduce the frequency and prevalence of diseases in the population. All of this work is ongoing and incredibly exciting. So as FUS expands, there's no question that around the world, interest is increasing. And it's really one of the most, in our view, one of the most exciting and important tools in the neurosciences. Much of the work has been driven by organizations like the Focused Ultrasound Foundation, which have driven a lot of innovation and novel work in this area. In close collaboration with industry has really been the driving force of a lot of this work, a lot of this novel work. So treatment centers are emerging in every corner of the globe, and they're emerging quickly. All the more reasons that centers like the ones at Utrecht are important. So these are going to be global centers of excellence that will help shape the field and push it forward for years to come. So with that, I'll thank the Focused Ultrasound Foundation and congratulate UMC Utrecht for their designation as a global center of excellence. Congratulations and thank you for your attention.
So, hello everybody, my name is Danis van Verde. I'm a pediatric neuro-oncologist in the Prinses Maxima Center for Pediatric Oncology here in Utrecht. It's the neighboring institute actually of the UMC Utrecht. And I want to present some of the preclinical data, but also the future studies we envision in pediatric neuro-oncology. So there is a clear need in pediatric and adult brain tumors, especially in high-grade glioma and low-grade glioma. So when looking at pediatric high-grade glioma, there are in general two big entities, the hemispherical and the diffuse midline gliomas, and these have a dismal prognosis. So 20 children in the Netherlands suffer from this disease, being 800 children in Europe, and hemispheric high-grade gliomas have a poor survival of around 20%. And for DIPG, which is located in the brainstem, there's no curative treatment, unfortunately. And after biopsy and radiotherapy, patients rapidly deteriorate and they die mostly within a year. And so this is the Kaplan-Meier, which shows this dismal prognosis. And these are three pictures of the same boy. So also during the disease course, patients are debilitated, also because of use of steroids, and their quality of life is really poor. Also in adults in high-grade glioma, being 960 adults in the Netherlands every year, there's also, these are largely incurable diseases. And the standard therapy in these patients is resection, radiochemotherapy, mostly with temozolomide, but also for this patient group, there is treatment innovation needed as most patients die also very soon. BBB disruption is largely heterogeneous, so there are enhancing regions in the tumor, but there are also large areas of the tumor where the blood-brain barrier is mostly intact, and these regions also need therapy. When looking at low-grade glioma, which is actually the most occurring tumor in children, these are mostly grade 1 optic pathway gliomas. And there are about 40 children every year in the Netherlands. These children suffer from progressive blindness. And therapies that we give to these children are mostly very long. So they take like 2 years of carboplatin and vincristine. And these therapies are mostly focused on disease stabilization. And most of the effects that we see are 25% reduction of tumor size, but these tumors rarely disappear. And in adults, low-grade glioma is slightly different. These are biologically different entities, 240 patients per year in the Netherlands, and these patients suffer from a shortened life expectancy, epilepsy, and chemotherapy mostly has limited effects due to an intact blood-brain barrier. Surgery and radiotherapy is the mainstay of therapy, but also these tumors suffer from malignant progression into high-grade gliomas, and these patients then mostly die. So we all know that our biggest enemy is the blood-brain barrier. It's very difficult to get drugs across, and we all know that small molecules are to some extent able to cross the blood-brain barrier, but most molecules that are bigger do not. So we do a lot of research in the laboratory, and there we see that these tumor cells of patients are actually quite sensitive to some of those drugs, but unfortunately these drugs do not cross the blood-brain barrier. So this is, I think, where focused ultrasound could play a very important role. So in my research group, we have a pipeline from bench to bedside. So we explore drugs in vitro in cell lines of patients. Then we try to mimic as much as possible the kinetics in patients. So we aim for a tissue AUC and looking how these drugs can work on tumor cells. Then we validate that in vivo, in animal models of this disease, so PDX models, as to ultimately try to have enough evidence to bring this into the clinic. So we've got several preclinical projects now running. The first one being a project where we focus on the effects of chemotherapy in PDX models combined with focused ultrasound. In the next project is a project where we look at radiation sensitization using drugs that sensitize tumor cells to radiation. So combined focused ultrasound with radiotherapy in PDX models. The third project is a project where we look at nanopackaging of drugs in combination with focused ultrasound, also in PDX models. And we have currently a project under development which looks at modulation of the local tumor microenvironment in combination with focused ultrasound. And I think what is a very interesting new avenue, and I think also Neil Lipsman hinted to that, is to look whether you can use focused ultrasound in the treatment of kids, looking at specific biomarkers. For example, in diffuse intrinsic pontine glioma and diffuse midline glioma, the histone mutation is in all the tumor cells, so that might be a very good biomarker to follow up with focused ultrasound and to see whether during therapy this changes. So I think with the establishment of the Center of Excellence, the UMC Utrecht, with the new focused ultrasound application for blood-brain barrier disruption, we have the possibility to, in the near future, have a very clear research field, clinical research field into pediatric neuro-oncology and adult neuro-oncology, and run phase 1 clinical trials in adult high-grade gliomas, but also low-grade gliomas, and looking at liquid biopsy studies in these patients, and ultimately expand it to other brain diseases such as neurodegenerative diseases and neuromodulation for epilepsy, for example, in kids and adults. So with that, I would like to thank everybody, and I think I'm really very enthusiastic in this collaboration of the Maxima and the UMC Utrecht, and I see a very bright future in this field. Thank you very much.
Thank you for the invitation. Today I'd like to talk about immunotherapy, which is the largest breakthrough in oncology in the last decade. And it is a major breakthrough because it has shown to induce really durable responses in patients with metastatic cancer. An example is Jimmy Carter here, who currently is the oldest former US president because of the immunotherapy that he got for his melanoma with brain metastasis 6 years ago. But also for patients, for example, with metastatic lung cancers, the patients that respond to immunotherapy live for many years without treatments, and that's really a breakthrough in oncology. Actually, our immune system can pretty well control tumor cells, but tumor cells develop ways to block the immune system. As you can anticipate in the left panel, where you see a tumor cell blocking a T cell of the immune system. And what we do with immunotherapy, with checkpoint inhibitors, is that we try to release the brake that the tumor cells put on the immune system. And if we release this brake, the T cells can again target the tumor cells and kill them. Like I said, this works really well in melanoma. Here you see waterfall plots. The patients below the x-axis are patients that are responding to the treatment. And you can see in melanoma, metastatic melanoma, this is the majority of patients. But for example, there's also when you look at lung cancer patients or patients with Hodgkin lymphoma. But then if you look at more common tumors, such as, for example, colon cancer or breast cancer or prostate cancer, you can see that only the minority of patients respond to the treatment. And we think this is the case because the tumor cells of these tumors have fewer mutations in their DNA, and they look more like healthy cells and are less well recognized by the immune system. And therefore, the immune system is less well capable to induce an immune response against the tumor, and immunotherapy works less well. This is our largest challenge for this research, trying to make these less immunogenic tumors visible for the immune system, empowering immunotherapy, and in therefore delivering effective anti-tumor treatment. We think that histotripsy might very well be capable to accomplish this. Histotripsy is a non-invasive focused ultrasound that can cause localized and rapid tissue fractionation. And as it is very precise, it leaves the vasculature and surrounding tissues intact and creates a very sterile inflammation that leads to the release of tumor antigens, which are small parts of the tumor that are different from healthy tissue, and leads to the release of immune stimulating substances. Avinash Iranki and colleagues at the NIH and Children's National in collaboration with Grit Mone at our institute studied the combination of histotripsy and immunotherapy in a neuroblastoma mouse model in which they injected the mice with neuroblastoma, then treated them with histotripsy, and after that with a combination immunotherapy. And as you can appreciate on the lower graph, the mice that received either histotripsy or immunotherapy alone, the blue and the red line, died within 1 month, while the mice that received a combination of immunotherapy and histotripsy lived much longer and had a very durable anti-tumor effect. And which was interesting was that this not only occurred in the tumor that was treated with histotripsy, but also in the tumors on the other side of the body of the mice. They also looked at the effects of histotripsy on the immune system. And in the left graph, they looked at the cytotoxic T cells, which are our heroes. These are the T cells that can kill the tumor cells, and they saw an increase after 24 hours. They also looked at the dendritic cells that tell the T cells where to go, and you can also see an increase there. And they looked at the Tregs, which kind of inhibit the anti-tumor response, and they saw a decrease after 2 and 3 days of treatment. And these were the effects of histotripsy alone without immunotherapy, which shows the effect of histotripsy alone on the immune system. We think that these mice data are really promising and therefore are preparing for a phase 1 study in humans, in which we include adult patients with metastatic or unresectable cancers that have no standard treatment options and at least one primary tumor or metastasis that is accessible for histotripsy. We'll first treat 3 patients with histotripsy alone, and then 6 patients with the combination, and after a safety stop, another 18 patients. The primary endpoint of this study is safety, and secondary endpoints, of course, we'll be looking at the immune response in the blood and in the tumor, and the response of the tumors. We think that the combination of histotripsy and immunotherapy could work in tumors that normally do not respond to immunotherapy. And this phase 1 study will assess the safety and explore the immune and anti-tumor responses. Due to time, I'd like to acknowledge all these people, but especially the FUS Foundation for promoting therapeutic ultrasound and the clinical translation of this. This was my presentation, and I'd be happy to take any questions.
Hi everyone, it's my pleasure today to talk about the further project. So before going into the detail of the project, I would like to give you a sense of what was the path that as a scientist, clinical scientist, I went through these years of using focused ultrasound for treatment of bone metastases. Particularly when I started, it was 2010, and patients' treatment was totally unknown to us. So I would like to give you a sense of what was the treatment in that days, showing you the very first patient that we enrolled. It took almost a month to understand which patient could fit the treatment, and it took about 10 days prior to the treatment itself. So between patient enrollment and the treatment, it lasted 10 days because we had to set up the MR unit since it was something that we were not used to it. So this patient was a patient with melanoma, with right scapular metastasis that was not responding to radiation or painkillers. So he had a pain score of 8 out of 10. And this is the treatment time. As you can see, the patient was under general anesthesia. This is the transducer. And treatment lasted 1 hour and 20 minutes with 26 sonication. Interesting for all of us was that the patient discontinued pain medication in 3 days. So pain effect was very quick. And this is something that was completely unknown to us. And with growing experience, the treatment became actually faster, and the number of patients treated was growing every day. And we were able to demonstrate the results of this treatment even in a phase 3 trial comparing MR-guided focused ultrasound against sham. And for example, this is a patient with pain score of 10, breast metastasis, lesion was not responding to radiation therapy. And again, very quick treatment, 45 minutes, and excellent result with the pain score dropping to 0 quickly. Here is another patient with breast cancer, but differently, this patient never received radiation therapy on this specific lesion. So we decided to treat also this patient, and the treatment effect was very enthusiastic actually, because the pain dropped to 0 in few days, and 2 months later, the lesion started to mineralize. That this is something that happens quite often in our population. We also grouped these patients that were treated as a primary pain palliation intent, and actually the results were very, very good, as you can see. So all this experience brought us to the need of a clinical trial, a phase 3 clinical trial that was going to compare the actual clinical performance of focused ultrasound against radiotherapy. So we studied a design entailing 3 different arms with a randomization 1:1:1 between standard radiotherapy, MR-guided focused ultrasound plus radiation, which is a very interesting group of patients, and also the effect of focused ultrasound itself. In case of patients not responding to focused ultrasound, they could undergo radiotherapy on a crossover fashion. So the project tried to respond if patient treated with MR-guided focused ultrasound will experience a faster early pain response in comparison to radiation therapy, with primary outcome of patient-reported pain response 14 days after completion of treatment. And 14 days is a fair balance between patient enrollment, patient treatment, and expected outcomes for both radiation therapy and focused ultrasound. So we expect to enroll 72 patients for each group in a total of 216 patients. And this is the geographical distribution of the centers participating in this project. By now, all centers had ethical committee approval, and some of us already started to enroll patients, but with the pandemic situation, most of us are delaying the first patient inclusion. So hope we can speed up during the next months. Thank you very much.
Good day, my name is Martijn Boomsma. I am a radiologist and the project leader of all MR-HIFU treatments in Isala. Thank you for this opportunity to present our experiences with MR-HIFU on this special, festive occasion. I'm proud to be able to present you our multidisciplinary team. At this moment we perform both uterine fibroid and painful bone metastasis treatments. Our team is amongst others supported by 3 PhD candidates, and in the following couple of minutes I will show you some glimpse of their beautiful. We collaborate with UMC Utrecht on different aspects. Amongst others, our PhD candidates in MR-HIFU attend the UMC Utrecht Graduate School Medical Imaging called IMAGO. This is a time span of 7 years in which we started off with MR-HIFU. From the beginning of closing MRA with Philips, it was clear that a collaboration with the UMCU would be beneficial for our implementation. Together, we will be more effective in adoption of MR-HIFU as a first-line modality treatment for uterine fibroids. Next, we performed the first MR-HIFU treatments of uterine fibroids while performing a study to improve the existing screening protocol for eligibility. Other elements of MR-HIFU could be improved afterwards, including manipulation, but not before we systematically evaluated the treatment effects of MR-HIFU, and by use of the data from the UMCU, we could investigate long-term treatment efficacy determined by re-intervention rate. Implementation of our developed 3-step modified manipulation protocol during MR-HIFU therapy of uterine fibroids improved the eligibility percentage and reduced the treatment failure rate. We are currently investigating a medical intervention with carbetocin, which is a uterine stimulant that has a dehydrating effect of the myoma and makes heating of the cells more effective. These data were discussed in detail at RSNA 20. One of the major issues in treating fibroids is the impossibility to evaluate the non-perfused volume during the treatment. Our multiparametric approach using paired data in all patients show promising results estimating non-perfused volume by using diffusion-weighted imaging. The use of deep learning algorithms, fed by paired data of both diffusion and post-IV gadolinium imaging, has given us the opportunity to reproduce non-perfused volume images without the use of gadolinium, and thus enables us to shorter procedure times and a more effective treatment. Wouldn't it be great if we were able to correlate multiparametric imaging with true histology? In this pilot study, we are able to correlate histological findings such as vessel density with multiparametric data from the screening MRI scans. In our opinion, this is the first step to start histological mapping by the use of MRI sequences. We have the intention to validate this work in tight collaboration with the University of Amsterdam and Oslo. After successfully implementing MR HIFU for uterine fibroid treatment, we became partner of the Horizon 2020 project. Together with the University Medical Center Utrecht, we organized and performed pre-Verde, and this was the jumpstart for us in treating bone metastasis. We investigated the safety and feasibility of a combined palliative bone metastasis treatment with MR HIFU and radiotherapy. We included 6 patients, and they all tolerated the combination treatment well. The treatment was safe and feasible. On average, pain declined 3.5 points on a numerical rating scale for pain. In total, response after 7 days was 83% and decreased to 60% after 4 weeks. Extended clinical outcomes of the combined treatment will be assessed in the ongoing Verde study. Verde is a RCT consisting of 3 arms for eligible patients for HIFU, on which my dear colleague Alessandro De Napoli will elaborate on this in his presentation. Fortunately, despite the COVID-19 pandemic, we were able to randomize and include the first 2 patients in ISALA. We are the principal investigator of the National Multicenter RCT MyChoice that will provide currently lacking information on the long-term effectiveness of MR HIFU for the treatment of uterine fibroids in comparison with standard care. The control group includes hysterectomy, myomectomy, and uterine artery embolizations. Follow-up will take 2 years, and our primary outcome is patient-based. For more information on the MyChoice study, please visit our study website mychoice.eu. At the moment, we are still on track in formalizing our partnership between ISALA and the UMCU. And well, actually, this is the most important slide of this presentation. From Zwolle to Utrecht, congratulations to all of you UMCU power people. We all in ISALA are so proud of you. Never stop exploring, up to infinity and beyond. Enjoy this beautiful day.
So in my 5 minutes, I would like to share with you our experience with the MR HIFU treatment of breast cancer at the University Medical Center of Utrecht. All the studies have been conducted with this team. We use a dedicated breast MR HIFU system that is in-house developed. As you can see on the images on the left, the transducers are placed in a cross surrounding the breast cup at a 15-degree angle tilted towards the chest wall, so we will be able to reach tumors that are located deeper in the breast. The spreading of the transducer surrounding the breast cup decreases the near and far field heating and therefore the complications. In the past, we have conducted a phase 1 clinical trial between 2012 and 2014. We have treated 10 patients with an invasive breast carcinoma. We used a treat and resect protocol. We did a partial tumor ablation to allow us to evaluate the accuracy and precision of the sonications. And the patients underwent conscious sedation during this HIFU treatment and were subsequently treated with standard therapy that can be breast conserving surgery or mastectomy. Our study endpoints for safety, feasibility, and the accuracy and precision of the sonications. On the picture, you can see a sonication in the breast of one of the patients. The results of the phase 1 clinical trial were that we had an ablation zone in the tumor in 6 out of 9 patients. In 1 patient, no surgery was performed because she refused. And in 2 patients, the ablation zone was located outside of the tumor in the fat because in 1 patient, the tumor could not be reached. And in the other patient, the focal point was off target. The ablation zones were sized 3 to 11 millimeters. While we used the 6 millimeter cells in all patients, the accuracy and precision were good, about 2 millimeters in each slice, coronal and sagittal. On the contrast-enhanced MRI performed directly after treatment, we did not see a non-perfused volume. This could be either due to the small ablation zones or due to the short interval between treatment and MR. The clinical results were excellent. Only 2 patients had local pain, and 2 patients had nausea from the sedation medication. In the picture, you can see the ablation zone in a breast tumor on macroscopy and microscopy. At present, we have 2 clinical trials for breast cancer. The first trial is the ablation trial. It's approved end of 2019, but currently on hold due to the COVID crisis. The study design will be that we hope to treat 10 patients with early breast cancer. Instead of the first phase 1 trial, we'll do a total tumor ablation with this HIFU system, followed by standard therapy to evaluate our accuracy and precision and effectivity. Our secondary endpoint will be safety. So on the lower half of the slide, you'll see the scheme used for the trial. We'll start with an MR to see if patients are suitable for therapy. Then we'll do the MR HIFU therapy, and within 7 days or after 7 days, we'll do an MR to evaluate the objective response and a medical photo to see how the cosmetic results are. And then the standard therapy will follow. The second trial we have is for newly diagnosed stage 4 breast cancer. It has been approved in the end of 2020. We hope to include 12 patients. These patients are normally treated with the AC scheme, including doxorubicin, to treat their primary tumor and distant metastases. The standard doxorubicin will be replaced with temperature-sensitive doxorubicin, also called thermodox, and we will induce hyperthermia, leading to a higher leakage of the doxorubicin in the heated part. So the patient will be positioned in the breast cup with the breast, and the hyperthermia will be induced by the MR HIFU system, leading to higher doses of doxorubicin in the primary tumor. And as you can see in the image, there's also leakage at 37 degrees. So the distant metastasis will also be treated by the normal leakage of the thermodox. This has proven to be equal to the normal peripheral dose of doxorubicin compared in the normal AC scheme. The endpoints of the study are safety, tolerability, and feasibility of this study. And the secondary endpoints are distant and local response rates. So this is the scheme for this study. There are normally 6 cycles of chemotherapy. These all will be replaced by hyperthermia and thermodox, followed by cyclophosphamide. We will start the hyperthermia. If this is stable, we'll start the infusion of thermodox and continue the hyperthermia for 60 minutes. We will evaluate our results after 2 cycles and all cycles. And if a complete response is established during one of the cycles, we will switch from the hyperthermia and thermodox to the normal scheme. Finally, I would like to share some thoughts on the future. We now have a study for stage 1 and stage 4 disease, stage 2 and 3 disease, which is about 50% of all patients, are currently treated with neoadjuvant chemotherapy to reduce the tumor size or treat axillary metastases. I hope in the future these patients will also benefit from either chemotherapy followed by ablation, as would be excellent for the patient on the left, which had a good response on chemotherapy, or chemotherapy can be replaced by hyperthermia and thermodox, which would be a better treatment for the patient on the right, which has had a poor result on the standard chemotherapy. Thank you very much.
Used my co-moderator here, Professor Peter Leuthen, who is the former head of the Department of Imaging and Oncology and now Vice Dean of Life Sciences at the University of Utrecht. Peter.
Thank you, Thomas. A real pleasure to be here, and also I want to congratulate my own colleagues as it is with this very nice accomplishment. In terms of starting off the discussion, is Karijn, are you still on?
Yes, I am.
Okay. I want to intrigued by all the promising things that you shared with us. Maybe you can elaborate a little bit on the application that you show, the combination of the histotripsy with immunotherapy. There are alternatives as well. Radiotherapy is one example. But if you would compare this with the other image-guided therapies, what do you expect, or which maybe do you hope is the advantage of this particular way of disturbing the system and creating this immune response as you compare it with other possibilities?
Yeah, I think that we think and hope that histotripsy gives a more targeted, sophisticated, controlled local inflammation. So there's less collateral damage, and you can, I think, better give a controlled immune inflicting
response.
Yeah. Response. And I think for other approaches, we're afraid that the response is too much. It gives too much damage also to the immune cells that you need in order to stimulate the immune response.
Okay. And what would be the preferred sequence and timing of those two treatment modalities? Are you doing it right one after the other, or is there some time lapse in between? How do you find out what the optimum will be?
Yeah, I think it's a really interesting question. In the mice, they first gave the histotripsy and then the checkpoint inhibition, but the half-life of the immunotherapy is pretty long, and you want the immune system to be ready for priming. So you want it to be ready once the neoantigens, so the part of the tumors that they need to recognize, come along. So we think that we need to start with the immunotherapy first and then introduce the histotripsy shortly after that.
Okay.
And that's also what they suggested in the clinical cancer research paper.
Thanks. Other comments of the panel members on this particular issues that I just raised, maybe? If not, then I have a question of the foundation's chair. Neil, are you still on the?
I am.
Okay. So maybe it's even a sort of well, I wouldn't say indecent question, but so what you see in this field, it is remarkable how many different applications are emerging, and maybe applications that maybe just a few years nobody had thought of. But having said that, and I know it's like this foundation is creating many different children, and you never ask a parent, which of those children you really favorite, but sometimes it's important to focus a little bit on those applications that really address an unmet need, or where there's really no alternative, to speed up the general acceptation of that technology. And we have seen a lot of applications, particularly in oncology, in this last hour, but there are even much more than that. What is your opinion on that?
Well, let me address it in a couple of ways. First of all, 10 years ago, there were only 3 clinical indications in various stages of research and development and commercialization. Today, there are more than 130, and the number is increasing and increasing dramatically. From the foundation's perspective, we're interested ecumenically in all of these disorders, but where our team concentrates our mental energy are on the CNS indications: Alzheimer's, Parkinson's, OCD, epilepsy, stroke, and so on, and cancer immunotherapy. So we think that this is going to have the largest impact. But it's a little bit of a tricky situation to figure out where to put our time and money and energy. For instance, 7 or 8 years ago, we decided to pursue essential tremor. Why? Essential tremor, in an epidemiological sense, is insignificant. but compared to prostate cancer and breast cancer. But why did we decide to pursue that? First of all, everyone is interested in brain indications. Everybody's interested in how the brain works and is concerned about getting brain disorders. They're the most feared disorder amongst patients. Number one. Number two is the fact that if the technology could work on something that is deep in the brain with a high degree of precision and accuracy in an awake patient without any incisions or holes in the skull, everybody would believe that that same technology could be more readily used on things that are outside of the body, like tumors of the breast or tumors in more forgiving locations, like the liver. And the third point is time. So the patient goes into the machine having been disabled by a central tremor for 10 years and comes out cured. It's a little different than treating a man who's an 80-year-old guy who's got an elevated PSA of 12 and then measuring his PSA a year later and it's 8. It's not as dramatic. So I would submit that had it not been for the visibility, the dramatic visibility that treatment of essential tremor created, this whole field would have not evolved nearly as rapidly and maybe would not have evolved at all. So that's a very long answer to what was seemingly a simple question. I apologize.
No, I mean, thank you for, I think it's very insightful how those things work, because it's not trivial at all to come forward with a complicated new technology and really, you know, get a broad level of support to bring it forward. Thank you. Thomas, yeah. I hand that over to you.
Yes. Okay, thank you. Now there's the brain application, blood-brain barrier opening that sparked a few questions, and one is I would call it almost the obvious. Can HIFU penetrate the bone of the skull without weakening attenuation of the focused beam? So maybe Dr. Lipsman, you can answer that one before we come to some more questions on blood-brain barrier.
Sure. No, thank you for that. In short answer, yes. And I think that, you know, when we talk about focused ultrasound for the brain, we're really distinguishing between two different kinds of focused ultrasound. So HIFU is high frequency, high intensity focused ultrasound, where the goal is really to generate a lesion in the brain. And at the intersection of the ultrasound waves is where tissue temperature is raised and you generate a lesion, basically thermocoagulative necrosis at that intersection point. When we're talking about low frequency ultrasound, we're talking about much lower powers. So orders of magnitude lower powers, about less than 1% of the power that is used to generate a lesion is used to open the blood-brain barrier. So temperature rises are much smaller. And as a result, we can access regions of the brain that ordinarily we cannot access with high frequency ultrasound because of adjacent heating of dura and brain and skull. So for both, the skull is important, but it's much less important for low frequency applications than it is for high frequency applications. So we're finding our flexibility in accessing different regions of the brain, even deep regions of the brain, brainstem regions, and even close to cortical surface, is much better with the low frequency drug delivery applications. I'll also add just one more brief comment to what Neil was saying about the power of this technology in neurosurgery, and that is that using ultrasound or attempting to use ultrasound to treat brain pathology has been a goal of neurosurgeons and neuroscientists for several decades, stretching back to the 1940s. And up until really the 1990s, it required cranial access. So it required a removal of part of the skull in order to do this. And it was really only in the last 20 or 25 years that the skull problem has been solved. And now we can do it totally non-invasively. But this, nevertheless, has been a major issue and a major goal of the neurosciences for several decades. And tremor did provide the first proof of concept for that. So it has a historical sort of meaning and significance for many of us.
Thank you. You talked about blood-brain barrier opening in oncology, primarily in oncology, but this is a very powerful mechanism of action. Do you see, or can you elaborate on other indications, applications for this mechanism of action?
Yeah, I mean, we're quite interested in several other platforms. I mean, we at Sunnybrook are fortunate to have, Kalervo Hinnanen, who's one of the pioneers of focused ultrasound physics and technology, as our close collaborator, and a robust physics, engineering, and clinical team working on several platforms. So we have the oncology projects. We're also very interested in neurodegenerative disease, where we're using focused ultrasound to open the blood-brain barrier in Alzheimer's. Leveraging preclinical data that suggests that there may be some efficacy there, as well as in Parkinson's disease, where we're hoping to deliver therapeutics to the brain as well. So those are, as I see, the two major fields that are poised to make a significant impact, or that focused ultrasound is poised to make a significant impact on the neurodegenerative diseases and oncology.
That sounds very promising. But one last question on blood-brain barrier opening. The blood-brain barrier is there for a reason. So is there a risk of opening that blood-brain barrier, even temporarily?
Yeah, I mean, that's a great question. Of course, it's there for a reason. Now, there's a lot of good things, obviously. It prevents a lot of large compounds from entering the brain, a lot of dangerous things from accessing the brain, but of course can prevent potentially effective therapeutics from entering as well. That's really the objective of a lot of these phase 1 trials, to demonstrate that it's safe to do so, that it's technically feasible to do so, that in fact it is reversible, that the blood-brain barrier closes, and that whatever effects we're seeing are transient. So we are very interested in looking at the local cellular and biochemical milieu around the area where you're opening the blood-brain barrier, looking at peripheral biomarkers of things like inflammation, neuronal and glial damage, and sort of looking at what exactly we're doing and how we can demonstrate that in fact it is safe to do so. But as a first step, it's critical, absolutely, to establish safety, and that's where a lot of our efforts have so far been geared.
Thank you. I have 2 questions for Dr. Brad on breast cancer. First one is, can you tell us about how the HIFU breast cancer patient that refused the resection did later on?
Good question. I'm sorry, I don't know. She came from another hospital and was referred to us on her question, and then she just disappeared out of follow-up. So I do not know how she did. It was only partial ablation, so I'm assuming that it did not work out that well, but depends on the normal biological behavior of her tumor. So I don't know.
Okay. That happens. Second question is, will the studies accept remote patients for treatment, or is it only patients from the Netherlands? Patients coming from France or from...
No, we haven't talked about it, and sometimes we are asked by patients from Belgium or France or Turkey if we want to do a HIFU treatment for something, for fibroids or for breast cancer. I don't know. I think it would not be a problem because we can still do the treatment, but they would have to have the surgery here. Otherwise, we won't have our endpoints. We don't know if the pathology, if it worked, and if it didn't work, they have to do the receptor status and everything again on the primary tumor. So it is important for the patient to have good pathology before the treatment, and if it's to determine what the adjuvant treatment should be. And if they don't have that, or if the tumor ablation is not fully, they should have it here all over again. So I don't know.
Okay. Well,
everything
also
should
for
be
the
done
follow-up, it would be. Right. And that's a lengthy process, right?
All is a lengthy process, including the HIFU treatment as of now.
Yes, right. Peter, do you have more questions?
Well, I have a question, something that wasn't really addressed, but that goes back to the CNS applications. And I presume Dr. Lipsman would be the person that we could answer that. I'm just interested in what the status is of the current applications in Alzheimer, because what we've seen is that a couple of years ago, a lot of phase 3 trials of Alzheimer drugs more or less failed. But you see, if I'm not mistaken, a real revival of those studies in patient groups that are in a much earlier stage of the disease, if not even in the prodromal phase, where there's no clinical symptoms, but we know because of genetics that those patients, well, they just can wait to develop Alzheimer's disease. So what is your take on that?
Yeah, no, that's a really great question, and there's a lot there to address. It definitely, the non-invasiveness of ultrasound and the fact that we can do this as an outpatient procedure lends itself better to earlier stages of the illness. But we needed to do the phase 1 and early phase trials to show, in fact, that opening the BBB in these patients in amyloid positive regions that are prone, of course, to bleeding and other issues is safe. Having that safety, pretty compelling safety data that we have so far, and then we've published, suggests that yes, we ought to be exploring this in earlier stages. The challenge with the Alzheimer's field, and this challenges basically all therapeutic trials, is which therapeutic to choose and which mechanism are you going to tackle. Of course, the amyloid cascade hypothesis is itself controversial, and the link between amyloid and pathology. Do you tackle anti-amyloid antibodies, which are expensive and requires close collaboration with pharma? Do you tackle tau in earlier stages of the illness and then look at sort of specific structures of the brain, medial temporal lobe and others, entorhinal cortex? Do you tackle inflammation? Do you look at specific genetic profiles of patients, as you mentioned, for example, ApoE4 homozygous patients who are more prone to amyloid? So there are a lot of levers to press, but I think that, arguably one of the most challenging things in Alzheimer's is going to be also outcomes and metrics. So what we look at, of course, is beyond safety. When we look at efficacy, we can define that in different ways. We can look at imaging efficacy, so improvements in amyloid and tau over time, with or without a therapeutic. But we've also seen some drugs be able to clear amyloid without a very clear signal on efficacy in terms of clinical outcomes. So then when you're looking at clinical outcomes, we're really looking at very long-term effects. So we have to wait several years in order to demonstrate, especially in prodromal and early cases. We know that amyloid develops sometimes 20 years before patients can get symptoms. So are we really going to wait that long in order to see efficacy? So as a field, we sort of have to design these trials that address efficacy in a very tricky disease like Alzheimer's disease, where the outcomes are going to be measured over years. So I'm optimistic. I'm optimistic that ultrasound gives you that kind of flexibility to try multiple therapeutics, novel trial designs that take advantage of all of these kinds of data that we're getting from all these trials to tackle it from different directions. But it will require sort of a global effort in order to make that happen.
Yeah. Oh, thank you. Thomas?
Yeah, we have, so acceptance of focused ultrasound of new applications in the medical community is often kind of, the question is about clinical evidence. So there is, Dr. Napoli has mentioned, there was a randomized trial for focused ultrasound for bone metastasis against sham that helped the application. And in other application, we still miss that. So now we have heard today of my choice from Dr. Boomsma and from further from Dr. Napoli, 2 randomized controlled trials that have a good potential to improve the acceptance and adoption of the field. In the past, such trials had always difficulties with patient recruitment and going then to the surgery part of the surgery arm of the trial. Dr. Boomsma, Dr. Napoli, how are you planning to overcome this and make sure you get the required recruitment for these trials? Maybe Dr. Boomsma first.
Thank you, Thomas. Well, that is the challenge. It takes a lot of dedication. lot of disappointments because always with an RCT there are a lot of restrictions when you select patients, and that's also the backdraft of an RCT, in my opinion. But nevertheless, it's the world we live in now, and we need to do these RCTs to prove insurance companies or the national institutes that are actually dealing with reimbursement or not to deliver this evidence. And of course for good reasons. But okay, so an RCT has a drawback, but the difficulty in recruiting patients is that you'll have to line up all the other specialties as well. So you have to have a very good relationship with your gynecologist, for example, for my choice. We had a slide in this one of the presentations. I don't really remember which one it was. It was about disruption. I think maybe it was from Lipsman. What disruption actually is, and we cannot underestimate the disruptive nature of a non-invasive treatment modality. Not just because of the substitution, but for the increased amount of patients that will be eligible for a treatment in an earlier stage of the burden of the disease. So other specialties might be a little bit reluctant to refer patients. They are also a little bit unknown with the treatment. There are a lot of, so that you need to inform them. You have to put a lot of effort in this. The consultation at the outward department of the gynecologist is of the utmost importance. That's the place where it happens. If the gynecologist does not think of HIFU to be considered in a portfolio, it won't even get the attention. So one of the things we are thinking of with the National Benign Gynecology Board of the Dutch Gynecology Society is maybe to make some sort of application or decision tool, so that when you sit down together with the patient in shared decision management, you actually have a tool that really can guide you through all the possibilities that are there with the right information. So this is something that's really important. And I know, for example, from Professor Verkooyen, that in Utrecht they have, maybe she can elaborate on that later on, but they have a special place where they send people to give them proper information about RCTs, because a lot of the time the clinicians don't have the time to actually elaborate a lot during the outward department visit to talk about all the possibilities, because we all have this vision of shared decision making, but it takes a lot of talking, and there's a large knowledge gap between the doctor and the patient, but even sometimes it can be the other way around, of course. So there's no magic bullet here or magic wand. We really have to put a lot of effort in all different kind of aspects of empowerment of the gynecologist, empowerment of the patients. And I think that is very crucial. And you need to have a very, very dedicated team of people. You really need to have, pit bulls roaming the outward departments looking for patients. They should not escape. Just kidding a little bit, but you have to be really on top of things.
Yeah. Sure. Okay. No, I understand. And it has always been a challenge, and I'm sure it is still a challenge. And we hope you will succeed in this. Dr. Napoli, how do you deal for this in the further trial randomizing focused ultrasound against radiation therapy?
Well, I echo Martin's words, since RCT is always a pain, so it will create problems in recruiting patients. Randomization will be definitely a problem. Now, we have a long history in Rome of treatments with focused ultrasound, and radiation therapists have been always involved. So we have a good team here, and we also are direct referral for our patients, meaning the patients look up for mark-guided focused ultrasound by themselves, and then we randomize the patients. So this may be also a shortcut for having a larger number of patients. Anyhow, it doesn't mean it will be easier than in another place. Still, RCT is going to be difficult. And specifically now that we are living this pandemic situation, we couldn't even start to enroll patients. So this is so it seems that obstacles are really multiple, and we need to face one by one. So we need to start having the access to the to a treatment like focused ultrasound available again, because by now, since our hospital is totally COVID oriented, so it seems that all other diseases, including oncology, are passed out, but this is not the case. But we run into politics afterwards. So what Martin was saying, that there is something that when you talk to the patients, when your experience, with your enthusiasm, patients got it, and they are very keen to elaborate a possible study, to enter a possible study. So that's something that actually worked pretty well in our department. So we are pretty much aligned with other centers in this further consortium.
And maybe one other thing, if I can comment on that, you have to have a realistic perspective on the amount of patient that your adherence of your hospital is, for example. In the beginning, for Verde, we looked up all the patients that were referred in 1 year for painful bone metastasis. There's about 660. And we identified on the existing protocol 80 patients that were suitable. But Alessandro and I are on a sort of hotline with WhatsApp and Silo, so we often talk about patients. And if you sometimes look, that there are some patients that are just not technically not, they cannot be treated. Although on paper they look eligible, technically sometimes they are not eligible because nerves are in the way, or you cannot treat enough percentage of the lesion to get some sort of positive result. So in my opinion, the low including amount of patients is always a problem with all these grants. I've heard that from, you know, the ZonMW and everybody. So if you have this realism while you're doing the RCT, and you immediately see that you're, even with COVID or without COVID, the referrals or the amount of eligible patients should be the same. But if you see it's too low, and actually we are seeing this as we speak, we should broaden the consortium very quickly. And that also is a lot of effort, you know, legally, documents, papers, blah, 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 blah. And actually the most important thing is that you get the radiotherapists on the National Radiotherapy Society on board. And the positive thing is that I have seen that I have, the positive side from doing research with the radiotherapist in this case is that I've seen the strength of radiotherapy. And in the beginning I was like, oh, there will be maybe a large substitution of patients. But I think there will be a special group of patients that are suitable, that can benefit. But the majority will still get the standard treatment, I'm sure, because you cannot treat all these lesions. So if you tell this story to the National Society, you take away their fear, you get them in, you get them on board, and make sure that MR-HIFU is just one of their tools. And I think that's also very important. It's not a play game or a PlayStation of the radiologists. It's their tool, it's their instrument. And whether we are treating them or they're treating them, we're treating them together. I think that's a very, very important message.
I think you're absolutely right. And that's a very good message or statement to conclude this seminar. We are running against the end of our time. It's a tool that is for not only for the radiologists, but for the clinicians, and the clinicians need to embrace it, and it's a collaborative effort. So with this, I want to conclude today's webinar. If your question was not answered, or if you would like more information, please visit our website, fusfoundation.org, or email us at info@fusfoundation.org. Thank you all for joining. Thank you for the panel. Thank you to the panelists, and congratulations again to UMC Utrecht for the Center of Excellence. And stay tuned to our newsletter and website for invitations to future webinars. Have a great day.
Congratulations.
Thank you for the thanks to the foundation too.